spines are flattened out a little bit, which is hopefully diagnostics for someone out there that knows about these sea urchins. Or actually, is this a heart urchin? Looks a little bit oblong.
It doesn't have very solid spines, which you typically see on.
Well, and you can see they're all like pointed backwards because of the current, maybe, or maybe not.
It's a bad spine day.
Oh, Sandra Brooks saying Areosoma. Oh, I guess the flattened portion at the end of the spines are poison sacs.
Hello, this is Chris Ma.
Hello.
Hey.
That is nice to hear from Japan, you.
Greetings from a post-return of Japan. So I just thought I would offer a quick ID on this urchin.
Please do.
Yes.
I'm not sure exactly which. I'm sorry?
I said, please do. Welcome.
Thank you. So I'm not sure exactly which genus and species it is, but this is any kind of therid. And I forget which one of you had made mention of the characteristic flattened end of the spine, but that's what I'm using to identify this as such. The tips of the walking spines have a little hoof-like end, almost like a little boot, and that's usually characteristic of these urchins as they walk. They're often identified in if there's such a thing as a common name for these deep-sea urchins, it's a pancake urchin because the water in the coelom evacuates when they're collected at the surface, and so when these are found, they're usually completely flat. The New Zealanders refer to these as tam o'shanter urchins. This particular one, I don't know if it's necessarily as poisonous as some of the others, but sea urchins in this group, even in the deep sea, excuse me, can be quite toxic or rather venomous, I guess is the correct word. And people that I've worked with have been stung by them. Apparently, the larger they are, the more treacherous they are. But we'll undoubtedly see more of these as we proceed, and probably with more sort of pronounced sort of hoof-shaped structures. And some of them in this area also have large kind of balloon-shaped structures emerging from the top, which some have hypothesized act as hypodermic needles. So these are really interesting as a group as a whole because they're considered to be kind of early stem group sea urchins. That is to say, they've been around for quite a long time. And these can be, in fact, one of the interesting little taxonomic trivia is that Echinotheroids were actually described from fossils before they were described from living examples. So they're interesting. Anyway, that's just my 2 cents since there's a story to be told. They're one of my favorite animals when we go looking at these videos. And I also would like to chime in probably a couple of days late and note that swimming sea brittle star we saw the other day was amazing. So
yeah, let's do definitely a quick zoom on that. Well, thanks so much, Chris. We are glad to have you back. We missed you on the first several dives.
I know I'm pretty far away.
Oh, not to worry. I'll make it the usual.
You'll make up for it.
And this is Stephanie, is it not?
Yeah. Hey, Chris.
It's good to talk to someone on the other end from the department. Welcome.
Yeah, people have been asking me about you, and I was like, I don't actually see him all that often. So.
Okay, great. Okay, let's come on.
They shouldn't have to.
Do you agree?
Well, anyway, give my regards to everyone, and I'm looking forward to seeing you've already seen so many interesting sea stars that I've only noticed in passing because I've been traveling. But I'm looking forward to the bonanza of interesting critters that you'll undoubtedly be encountering.
We are. Cool. Gonna do 30 meters at 0.15 degrees, speed decimal 2.
We got asked to look at one of the corals to the left, but I don't know how. Sorry, I just saw this, so I don't know what direction we were pointing.
One of those corals to the left.
Oh, any one of them?
Any one of them.
I guess.
Just to zoom in on them. Rachel.
Pilot, this is Watch.
You're talking
Go ahead.
About.
Hey, we got a request to look at one of these corals, so pick your favorite.
I would assume the one's right in the center.
Yeah, I'm assuming the
ROV
soft
now
corals.
bridge, move initiated.
And we have a request for collection. The white ones. I see several white ones. Rachel, you'll have to be a little more specific. Are you talking about the ones, the larger ones?
Oh,
Not
yes.
that one. So we're just reading on
Pilot,
the
this
chat
is
room.
watch.
Yeah, we're standing by.
Yeah.
Not this one, apparently.
Okay, so
But a white one.
so don't pick my favorite one, huh?
Yes, sorry, I didn't know.
No, that's quite all right. We're just standing by. Let us know which one you want to see.
The one we're looking at now, question mark. Sorry, the one behind, you'll have to say, because we're moving. And do you have any delay? I don't think you have a delay on your screen, Rachel.
The one in the background, that tall white one?
Sorry for the confusion, guys. I'm not sure if we have a delay here or not. Actually, it is the one that we can see right now just to the center right.
Oh, okay.
That's kind of fluffy and going down to the, it's almost
Pilot, this is watch.
ready to be out of view.
Yep, go ahead and watch it. Okay. I think I heard we were just driving off screen, though. Is that what you heard, video?
Rachel, are you still there? Is it
We'll
the one
sit here
in the
with
center?
the lasers on it. See if it's this
Yes,
one or
that one will work. Either of those, actually, which...
Let's go partial on
Yes,
this one.
that the lasers are on.
Yes. Uh-huh. Yes.
Great.
Thank you. There is a slight delay now that we're looking at we've got two different screens going, so,
Okay.
yes, that one.
It's a bamboo coral.
Actually, that's just a regular bamboo coral. That's not one of them. Thank you for
So
zooming in, though.
that's
It's hard
not
to
what you
tell
wanted?
from that far away.
Rachel, so you don't need a collection of that?
No, that's just a bamboo coral.
Okay.
There was one that we zoomed in earlier that looked
All right, pilot,
like
we can move on from
bamboo
this
coral
one.
Roger that.
far away,
Video, let's do a
but is not when you get up on
quick
it. So
snap
no,
on this
no, we
lower
don't
one.
need this
Make
one.
sure
Sorry.
this isn't it either.
Okay.
We've seen it several times, and we haven't been able to ID the one we're talking about.
All right.
But
And this is another
Yeah,
bamboo.
we'll
Another
try
bamboo.
and find another one.
Okay,
Okay.
let me stabilize my shot.
Fluffier
Hold
one.
the zoom. So it's definitely not this one either. Okay.
Correct.
All right, we're moving on then.
All right.
All right.
Can you take a quick look at the sponge on the right? That looks,
The largest one?
yeah, the large
Yep.
one.
Video, let's go. Just from here. I'll try moving this a little closer. As I come around.
Looks a little more deflated than some of the ones we've seen.
Come down.
I don't think we have any of our...
Oh, that's nice. This darker globular one, too.
Yeah, we
Spherical?
saw larger of those earlier, but that's very rounded, the one just below the lasers.
Pilot, this is watch one.
Go ahead.
Can we just take a look at that coral since we're here?
Thought you might ask since it was white and almost bamboo-like. Let's come wide. Video. Let me
And we've just left
set up a
little
bit better.
depression area that had very few corals and mostly sediment. And Scott Harris, one of the geologists in the chat room from the College of Charleston,
Okay, let's
says
go back
it's
in,
nice to see
please.
the sediment accumulating downdrift as expected, with the corals creating the baffle to help the mounds build. It's almost poetic, Scott. But very true. As soon as you get off these mounds, you see the quieter areas
way back.
between the mounds,
Pushing
and
back in.
the current is must be less current there, and
let's come
you're able
wide.
to
I'm
get some
Not going to be
sedimentation
able to hold it
material.
steady.
There's some of the anamastis. I can't say it.
Anthomastus.
Anthomastis. I have to practice at night, I swear. I'm learning all these names that I've never heard.
Okay,
The only reason I know that one is because we used to collect it in Monterey Bay, so
one more
I
try
have
video.
done my practicing.
That's great. So is this a black coral, perhaps? Did you already say that?
I didn't say that, but
Okay.
I agree.
I agree, yeah.
And we're not hearing people shouting that this is the one they want, so.
Okay, carrying on. Thanks, video. Sorry that took a couple attempts.
And can we get a quick view of the Anthomastus on the right? The red ones? Yeah.
You got it? Go ahead, video. Let's do partial. Yeah, hold there.
I've never seen
Beautiful.
ones that are all that deep red before. I'm used to seeing pink ones.
Yeah, very pink, yes, farther out.
I don't know if that means it's a different species of Anthomastus or if they just have color variability.
The bamboo coral in the front.
Yeah.
Or is this tall stalked, is that a...
I think that's a hydroid. Yeah. Multiple different kinds, or at least morphologies of sponges around.
Slowly drifting in.
And it looks like some hard coral, a little bit of hard coral
The orange
between
material.
the Anthomastus.
Really gives you a sense of how important the dead coral structure is to becoming habitat for so many other different organisms.
Is that good?
Especially,
Watch leads?
Yes, it is good.
All right.
Thanks, Pilot.
Just coming wide.
Oh, Nolan Barrett, so both those sponges, the spherical one and the tabular one, look like geodia,
Time for me to move on.
but different species.
All right. Thanks, Nolan. And we have another golden crab.
Thank you, co-pilot.
We've seen, I think, 3 of these today. This is clearly a habitat in which they live.
We look a little dark, video. Thank you.
Nice glass sponge there. What about?
Yeah, Sirius should be just about done swinging too, so should be too much more movement.
Gonna see if I can push out.
ROV nav bridge move complete.
They must be having quite a bit of delay because by the time I see the chat, I'm not sure what organism they're talking about.
I know that Scott France was having a 7-second delay, and I don't know if that's also
There's
what
quite
Rachel's
a
group
bit
is
of current here.
is
I'm
finding.
hard over on my laterals.
Look at all those
pushing forward.
eels, tons of them.
Slow moving. Okay, let's give a look off to our port, see what's over here. You can see how quickly.
Oh, that looks like one of those codlings.
We've seen several of the
How quickly I just start
They're
moving
very
if I go off
common
stick.
here, it seems, and we also saw them a lot in the Charleston Bump area.
Okay. And there's some.
So watch
nice
these. If you look over
colonies.
at Sirius, I'm off stick right now, so that gives you a sense of how quickly I'm getting pushed by this current. It's quite
You're getting
fast
pushed
down
laterally.
here.
Is that what you mean?
Yeah.
Yeah, how quickly I'm just moving across
Yeah.
the screen. Usually it takes much, much longer for that to happen. So there's quite a bit of current down here,
We don't have a lot
for
of
sure.
time to settle down
Well,
and take a look, but
It's not so much that, it's just, you know, when we're not fighting, there's a lot of current, you know. But we have time to get the looks you want.
okay, great, thanks.
Gonna push out, see what's on this edge up here.
Sure, we like zigzagging across these high points.
Is that the Analopsamia?
Analopsamia.
Is that
the
common?
Analopsamia is the stony coral that's most common around here.
Decide where we want to go after this, but it looks like we're at another high point, and we're very close to waypoint 3.
Okay.
Oh, you really see how mound-like the
I'm not
mound
seeing
is.
more
I
upward
really didn't
slope
expect
on
to see that
the
much
sonar.
of
Okay.
curvature at the top.
Yeah,
Zoom
exactly.
out
but-
sonars.
Go ahead. At least on D2, you're fine.
Great. Put both the 50.
Is that a? Oh, never mind. I'm seeing
So,
things.
Copilot, I'm going to run off a little bit and see just if I can shine my light beyond this edge here. But yeah, that might be the far edge there, Sean. I'm looking out east.
You're looking east, yeah, which makes sense.
So we might want to continue to move that direction. Because current looks like it's coming in, so I bet you know, over on that edge, we're going to get some good stuff as well.
Copy. Put you right. Stand by.
So, I mean, the 26 or whatever it was you did last time, I think will be fine.
Yeah, copy. I'll just repeat then. It'll be 30 meters at 015.
Sounds good to me.
Just get you to the edge, and then we can just kind of travel the edge up to
Exactly.
the next waypoint.
Yep. I'm backing towards you,
Is
Copilot.
bridge over?
Hey, ready for another bit?
Yeah, that'd be awesome.
Decimal 2 knots.
Okay, 30 meters at 015 degrees, speed decimal 2. Thank you.
Great.
So this is looking
Thank you.
east.
So we've made some good headway.
It's interesting that the top here is not very thickly covered.
This
It
is
was
very
kind of
different.
this. The patch we just passed is on the slope as opposed to the stop at the top.
That is unusual. It's almost as though the currents are streamlining the top. Maybe the current's too strong at the top.
Okay.
ROV nav bridge, move initiated.
And unlike previous dives where the organisms were attaching to rocky substrate,
See what's
which
over
in most
hard
cases
left.
would be more stable in a strong current, these organisms are attaching to dead stalks of,
Yeah, we don't want to go that way.
coral rubble,
It's down,
of the
down
stony
slope.
corals that have died throughout
So we want
thousands
to keep
of
going
years
this way.
and
All right.
left their remains.
Coming back
And
down.
most of that is now encrusted with an iron manganese coating that is from
Roger.
dissolved
let's
material.
hold.
Ions in the seawater precipitate out of the solution.
I'm going to try to
And
stay where we are
molecule
so
by
we
molecule
keep that
encrust
1.5 heading.
Just like
whatever is on the surface of the seabed. Yeah, you can see piles of the lighter color that's probably more recently dead. Hasn't been there long enough in the upper left screen. So the darker the coral rubble, the longer it has likely been on the seafloor. And of course, the living coral is bright white. And up ahead, we see another of those pancake urchins that Chris Ma mentioned, had educated us about their different characteristics.
and why they're called pancake urchins.
Yes, I can understand that now. And I'm
So-
glad we're not going to collect one since they have venom or they're toxic.
Yeah, venomous spines. Don't think our gloves are going to protect us
I
from
don't
that.
think, no.
Considering I was getting sponge spicules in my hands even with gloves on.
Great. And we have Cheryl back. Gonna allow Stephanie to go to lunch. Cheryl, this probably looks a little different from when you left.
Yeah, it sure does. What happened?
Well, we kept moving. We crossed a low spot that had almost no coral with lots of sediment, and then we saw some on the edge of this feature, but as we've climbed up, we've seen fewer and fewer coral clusters.
It's like there's a lot more dead coral here, or a total covering of the sediment by dead coral.
Almost looks like it's armoring the surface here, doesn't it?
Yeah, it does.
Just below center, we see another of the pancake urchins. Cheryl, we had Chris Ma join us for the first time on the expedition. He's back at the Smithsonian.
Oh, that's great.
He tuned in just in time to talk about the pancake urchin. So we got some good video of that.
Oh, that's great.
This mound is one of hundreds that occur across the region that we refer to as the
about
Mason,
175 since we're at the bottom.
Stetson Mesa, which has a water depth in the range of 700, 750 meters.
Yeah.
And it's a broad, flat area off the coasts of Florida and Georgia. It is beyond the continental shelf. It's a
we were told
lower
at 600
region
meters there's still a knot of current going northish.
Material,
so I think that's what's preventing the layback from
under the current of the Gulf
getting
Stream
any smaller.
And sits on the western edge of what we call the Blake Plateau. Our entire expedition has been to explore many different regions of this huge area called the Blake Plateau. And later on in the expedition, we'll move northward and see more of a characteristic continental margin off of the North Carolina coast. We have in this region, South Carolina, Florida, and Georgia, that stretches hundreds of kilometers off from the coast at a very flat region with water depths gradually dipping from about 400 meters to 1,200 meters. And then it cascades over into the deep waters at the Lake Escarpment.
It is quite strong.
And we were at the Lake Escarpment on our first dive several days ago, and then 2 days ago, and saw rock outcrops where the currents have kept the rocks clean and allowed for huge and diverse communities of coral. It was 2 days ago was just incredible.
Sure was.
Yeah, every view had hundreds of corals and many, many different species. So it was a lot of fun. Hopefully all of you will get to see video clips and photographs of that online in the next few days. We also went beyond the Blake Plateau and beyond the escarpment to a large area
we have
called the
a lot of
Blake
current
Ridge.
today. You can see it blowing by on my screen.
Lots of current.
So we just need to keep a close eye on the tether. As I'm turning around, we don't want it like blowing onto the vehicle, obviously. But basically, we're just kind of making our way north. They want to keep along pretty much this contour and follow the top of the feature. So we'll do the best we can here.
We have a pilot change here, so we're just settling out a little bit.
Copy. What's up? Yes. Yep. I'm just trying to get a feel for it here. It's really strong. Yep.
pilots are commenting on how strong the current is here.
So they've been going at about 0.15, so I think you can turn your head a little bit to overcompensate. So obviously you're 10 degrees off from that.
This is bridge over.
I just wanted to make sure, are we still moving?
No, we came to a stop.
Copy that, thank you.
So before we get started, I just want to figure out, you know, we need to think about what we would do if the ship did lose propulsion, seeing that we only have 3 generators operational, and we have had a loss due to the sargassum already this cruise. So okay.
While the pilots are
we would,
discussing the strategy in this very strong current,
yep. So Sirius is already pretty much
you want to
facing
compare
north.
this
So
area
I'll just
to
face
where we
south and
started
push
the
out,
dive, which
get
I
into
believe
tow.
was in a lower
That's all I need to
sheltered
do.
area of this current flow, because we weren't having such difficulties, and in that area we saw
Moving
huge
north. So
diversity
Sirius is
and
already
quite
facing north,
quite
and I would need to
an
stream
abundance
to the
of
south.
different corals and sponges. And now that we're into this much higher flow, we don't see that. So
But I'm
really
assuming that we
wondering
lose
if
ship
it's too strong
propulsion
a
and the
current
ship
for
starts
them here.
drifting 3 knots to the north, right? So I would just need to turn to the south as
What are
quick
your
as I
thoughts
can
on
and stretch
that,
out.
Cheryl?
Right?
Yeah,
Makes sense? Okay. All right.
I
coming down.
wouldn't have thought that too much current would be a problem, but who knows what they experience down
Yeah.
here, and this is just one little snapshot of what they experience. It could be even stronger at times, and
All right. So the ship is stopped.
but obviously there's been a lot of coral here over time. When you look
Certainly.
at the cover,
Yep. Okay.
hardly
So we
see
just
any
we get up
sediment. You see mostly just
maybe to
coral
like
rubble.
670 or something. Maybe we'd shoot for 650
We're
just to be
now at
safe.
700 meters. We started out
Okay.
this dive at more like 730 meters or so. So we've climbed about 30 meters over
Copy.
over 100 meters of
Okay.
horizontal distance.
Okay.
Yeah, I'm not sure what our
All right.
current location is
Yep.
relative to
Do
where we
that.
started.
So the last 2 moves have been at
You
0.15.
see that on
right? Okay. So pretty much
high pack
just a
view.
little off where Sirius is facing now.
the? Can't see that far.
Yeah,
So I'm going to come down to the bottom here. Levi can get himself sorted out.
quite
I was
read it.
comfortable.
It looks like
I'm going to
we
have to stay
are
facing about this
kind of on
direction
the
to get any
right
shots.
side of our second mound that we were going to climb up. So the biggest mound still lies ahead of us to the north.
And into faster currents. So we
Good.
may not get there. Seems to be increasing as we rise up these beaches.
All right. Watch lead. This is pilot.
Seeing some sponges again here.
Watch lead. This is pilot.
Go ahead, pilot.
Yeah, we're about settled in. Have you guys done a current test already? And if not, are you interested in doing that to get a feel for the speed here?
We have not done that, and that probably would be a good idea to get a feel for the speed, the current speed.
Okay. Can do that now. It's going to be quick.
We're doing a drift test to get an idea, a better idea of the
All right, you ready,
actual
Fernando?
current speed.
Okay. I'm going to go ahead and let off the stick now.
I have a feeling as we continue to climb up this, we're going to see more coral.
Yeah, it looks almost like in the distance there's some. Could be wishful thinking, hopeful.
And what's the resultant there? Speed norm, right? Isn't that 0.5 knots? Yep. Yeah, watch lead. So we've got about half a knot going from just south of east to going toward just north of west.
Hi, Cheryl. This is Chip Collier with the South Atlantic Fishery Management Council.
Hello, Chip. How are you?
I'm doing well. How are you
Thanks,
doing today?
Pilot.
I'm doing fine. Have you been watching our dive?
I've been watching parts of it, yeah. This is really
All right,
interesting
should we go ahead and
stuff,
get a move in?
We wanted to thank you guys for going out here.
Yes, thanks for suggesting
Yeah, I guess
it.
so.
Can
We'll
you tell us a little bit more about what you wanted to learn from this dive and from this area?
All right, any interest in anything here,
This area
Watchley?
was an important area that was identified back in 2008 through a mapping survey where they identified some of these mounds.
Any interest in looking at anything here, Watchley?
Mounds area.
I think let's just keep going a little bit.
Okay.
These
Thanks.
as the potential features of coral. And given some of the new Magnuson Act legislation language, that they wanted to protect these deepwater coral habitats. And so they established the Stetson Miami Terrace Coral Habitat Area of Particular Concern, which was enacted in 2010.
Bridge over.
And so it looks like this is actually
Okay, that's
very good
20
habitat that's being
meters,
protected.
Bearing 015, speed decimal 2. Thank you.
Well, excellent. So we did something right.
Yes, definitely.
I'm going to see if I can
That is good to hear.
At least 3
turn
out of the
somewhat
million
into the
mounds
slope here.
look like they're very good coral habitat, or 2 that we've looked at so far.
All right. Yeah, this is great information. I was watching earlier. I saw some golden crab on here, and those are some of the species that we are, or the council is interested in seeing where they live, and maybe some of their distribution in comparison to coral habitat as well as the mud bottom habitat. The fishermen have identified that the golden crab are actually more likely
ROV
to
nav
occur
bridge,
in
move initiated.
mud habitat than coral habitat, and so they try to avoid the coral habitat when they're setting their traps. Not only would it be bad for habitat, but it's also bad for their gear. Their gear could get wrapped up in it, so they actively try to avoid coral areas. So even though there's some golden crab in there, that's definitely not the primary habitat for golden crab.
Yeah, that's good to know. And I guess we won't be exploring too much mud habitat unless that's what we find in the place. And and then if we can get information about the
Look at that fish.
golden crabs, we will do that as
Cruising
well.
along.
Okay.
Are there any other species that are commercially fished in this area?
Probably not at this depth. Maybe royal red shrimp would be a little bit shallower than this, but that's not really a federally managed species. And then another species that might be occurring at some of these depths might be wreckfish. That's federally managed. I'm sure there's other species that are out there, but there's very few species that we manage at this depth.
Good to know.
We just saw another of the
Actually, one
cutthroat
species
eels.
we manage are the corals, and so we do have a fishery management plan for
Is he
corals.
just doing a bio
As
break?
odd as that might sound, we do try to protect them. They are very important habitat for a variety of species that we do manage. So.
We've been noticing that. Not that there's been all that many managed species, but they certainly do create a lot of habitat for all sorts of other invertebrates and
I'm just
and
going to
fishes.
hang out here on this patch and just circle around a little bit.
Yep, and
what's been happening
that
on
definitely
the
works
moves?
its way up the food chain. Those smaller
So you're pretty low to the
individuals are
ground.
likely to be food for the larger individuals.
There it is.
Right, it's all connected.
Okay, great.
But not much heave today, so that's good.
So what your layback is now.
Well, thanks so much for calling in and watching our dive today.
This is really exciting stuff.
Still not good.
Yeah, so we're going to be using information from dives like this to, one, confirm that some of these areas that we are protecting are, in fact, coral.
I've never landed with
we'll
that
use
much
some of the
layback
mapping to potentially indicate where other
Especially
coral
this
areas
shallow.
are.
No, it was absolutely
and then
insane.
some other products that might come out of this are going to be habitat
That worked out
predictability
great.
maps.
All right.
And they can for information like this.
We're waiting for the
and
ship to
combine
catch
it
up, but I
with
think we're
modeling
pretty well
and
set
figure
up.
out
as we
areas
move.
where
So,
coral are
video, do you want to
likely,
zoom in a
and
little
we
bit
can protect
and
those
see if
areas
we
as
see
well.
anything interesting here?
Great. Well, we did some nice mapping yesterday too when we weren't able to
How much better are
dive.
you
So
feeling
even
down
when
there,
we're
Violet?
About
not
half a
looking at
knot.
the seafloor, we are mapping it and trying to gather data that will help managers such as the South Atlantic Council.
Go ahead, watch
We
lead.
certainly appreciate any data we can get.
Yeah, the area is so vast that there's so
I'm sorry,
much
can
to
you
map
repeat?
and so much to look at that any chance we get, we will do what we can.
Okay.
And try to
Let's come out video.
just, solve some of the mystery of the area. It's so vast that there's a lot
does
of
not.
places
Well, wait.
we can't look
No.
at,
Okay.
but we can
It does not.
try to, like you said, use the habitat suitability
nav,
models
bridge, move
and the mapping to try to predict where there might be more
I see
habitat
it.
like
Okay.
this that we may not be able to look at right
So if
away,
it continues
but
on this trend, Copilot, I agree with
are
what he's
likely to
saying
be
there.
coral
It's like
habitats.
current to the northwest.
Yeah, and I mean, even these
Yeah.
areas where, I mean, not all the coral's living,
But no
it's
readings.
still important habitat, and, you know, that area can get destroyed by gear and still needs to be protected. So new coral can actually latch onto it and begin forming a new and larger reef.
Yeah, we've been noticing all of the species that utilize the dead coral, the scleractinian coral structure. It's just amazing. You can't even like count, and the more you zoom in, the more species you see. And then, like you said, other corals can grow on top of that matrix and create new
We were
reefs,
descending at 100%
new live
thrust earlier.
reefs. So yeah, it is quite a cycle.
The entire way
and
down. 15
this
meters per minute on the
habitat is definitely fragile and could be destroyed
1.5,
very
yep.
quickly if we don't protect it.
Okay, we can get lasers off.
So we're about
All right,
to
you want to
zoom
zoom in
on
video?
a pencil urchin, and the laser beams that are 10 centimeters apart gave us a sense of how large the body of this is. It's only a few
true.
centimeters
Oh my gosh.
in width.
Do not have a good footing here. Hold on.
Interesting that one of the spines is red.
Yeah, I've seen that many times before,
Really? I have
and
not. Huh.
it's funny. Just one
It's like,
spine will
guess
be
I'll just
red.
have to touch down with my
Does
left
it
toe and
eventually
not the
turn
right.
white? Is it anyone I...
This
I
clears.
don't know. I don't think so. But yeah, these are Sideris is the genus that is most common of these pencil urchins.
Yeah, it's not going to work.
Yesterday I was having trouble getting it to clear until I moved my right toe.
A little bit stirred up, but
Well, it's moving starboard to port, so when I touch my right toe down, it kicks up dust.
Yep. That's the same as yesterday, or 2 days ago.
Oh, man. Let's come out video.
All right.
looks like we might have had an anthomastus back there and several cup corals on this coral matrix.
Go ahead and go in.
Dead coral
we'll go
matrix.
from here.
Sponge and some more of that Duvia florida, which is soft coral, the brownish one up to the left. We have hydroids just above this pencil urchin.
It's like a brittle star hiding among the others. You can see the one leg in the back.
Gosh, look at that. This is just crazy. All right, let's come out.
All right, great, thank you.
So that's not even my thruster wash. That's just the current going by.
Wow. Yes, definitely the strongest current we've encountered on the expedition.
Well fed. Okay. I'm going to tilt up video and you can come back out a touch.
It looked like we had another bamboo coral there. More sponges. Another cutthroat eel just swam by. Pilot, this is watch one.
Go
At
ahead.
one point we had about
If we could
7
get a
cutthroat
zoom on
eels
one of
in
these
the view.
white corals,
they're
just
Sure.
to
very
make sure we
common
can
around
verify
here.
the species, please.
Absolutely. I see one on the left that will be a
A
good
very
candidate.
pointed I know they don't have
Like the ship is
noses.
stopped,
What would
Fernando?
you call the
Is that
front
correct?
of the cutthroat eel? Very pointed.
This is bridge over.
Snouts. They don't breathe. But you could easily imagine
we just
how they
arrived
could
at that location.
put their nose down into these corals in between the rubble and in between all the stalks.
Copy. You could probably do another 20
thread
if
their
you
way
want.
through these branches.
1-5.
So we're just taking a quick look at what the white coral is. We've been seeing both Lophelia pertusa and more commonly Analopsamia profunda.
Hold there.
We wanted to verify, and this looks like Analopsamia to me.
Okay, that's 20
Going
meters.
tighter.
Let's confirm that bearing of 015 at a speed of decimal 2. Thank you.
Cheryl, this is Chip. How are you
Levi, could
identifying
you turn
between
the
those
bridge
two different
volume down on
species?
my RTS?
Okay, that's a great
quickly?
question, Chip. So if you see on the back
Just like
left to
-12
the
or
dead
something.
coral matrix,
Thank you.
the Analopsamia has a really kind of small calyx
This is
that
bridge over.
doesn't have
That's
a
better,
lot
thanks.
of septa that are very that are sticking out towards us. So they have a more compact and not as much structure within the calyx.
ROV now,
So
bridge
that's a
move
really good
initiated.
way to tell. Lophelia will have bigger calyces with septa that you can see sticking out very prominently. So it's hard to tell on the actual live coral because the polyps are getting in the way of the structures that would distinguish the species, but on the dead coral you can see that.
Is that a squat lobster down in there?
Yeah, it looks like a squat lobster, the orange little chela that's sticking out there on either side, and a crinoid in the bottom part of the view. And that soft coral in the upper right is that Duva florida that we've seen many times. Seems to be pretty common around here.
Yeah. Yep.
Well, you can really see the current here.
Yeah. Everybody's
come out
hanging
a bit.
on.
There's a fish. Oh, he's gone.
We saw the fish shadow.
See how fast? Yeah. All right.
You're riding that train.
Did you need another view, or is that fine
No, this
for?
is good.
Okay.
Thank you.
Chuck Messing pointed out that the Sideris pencil urchin that we were looking at before may be the species Abyssocola. So Sideris abyssocola.
I'll try to have an altitude of no less than 10 meters.
We're still having some debate in the chat room, the
Copy.
scientists that are in the chat room here, about the type of bamboo coral that we've been seeing.
As the ship moves,
Scott France has been looking
my
through his database
altitude
of bamboo
drops.
coral samples,
and then as the ship stops, then
they've
my
found
altitude comes
in
back up a little bit.
this general area and depth
Yeah,
range
a little bit, I guess.
from 632
I've noticed
to
it, like,
687 meters.
a couple meters.
They were
maybe.
collecting a declayed keratoisis, but from shallower, 437 to 530 meters, they collected an
Just gonna
S-clade
kind of saddle back to you.
cladorisis.
Yep.
Sounds good.
And a couple of images he has available suggest that the
What was that
bamboo coral that we've
sample?
been seeing,
Port outer?
almost nodal branching with the sparse branches, looks like the cladorisis type.
Wonder if it would be easy for samples.
But
to
he doesn't have any images of keratoisis
Definitely.
in front of him
Fly out.
to
Definitely.
compare.
Probably want your face into the current then, right?
So Scott, if you would like us to
But
collect
then
one
we'd
of
be-
these
then
so
we'd be
that you
facing
can
downslope.
actually get a better ID on it, just let us
Yeah.
know.
And then if we turn against the current, the tether's going to be blown into the vehicle. So just have to
Figure it out.
figure it out when we get there. Yeah. Yeah, I'm running 90% thrust here. 100% on my lot. Can you help me keep an eye for motor faults?
Yeah, sure.
ROV nav bridge move complete.
Copy bridge.
There's a big
More
coral up
complete.
there, right? See what I'm seeing?
Yeah. Possible fan.
Yeah, we're moving that way. We'll be there shortly.
So that move was complete.
Looks like we have a larger
Yeah, let's do
octocoral on the
another
horizon here
one.
coming
Yeah.
up.
a good direction. 1-5.
Yeah, agreed.
Maybe the one after that might change
You
to
can
like
see that
due north.
there
But
is all
Definitely want to
sorts
get to
of
that big coral
dead
up there.
coral
Yeah.
rubble that is covering the sediment
Right,
on this
this is
part
now.
of the mound.
Bridge over.
Yeah, I was just
Let's
talking
keep
with our
moving,
mapping lead,
same as
Derek
before,
Sowers,
20
and
meters,
asked him
bearing
what the
015,
backscatter signal
speed
is
0
in this
2.
area.
Okay, 20 meters, 015
And
degrees.
backscatter
tilt down on your
is
camera a
a
little bit.
measure of how much of the sound used to
Good copy.
from the multi-beam sonar,
Yeah.
which we use to determine the depths. We can also measure the intensity of the signal as it returns to the ship, and the variation in the intensity gives us a first
yeah, if you wanted to turn more, I could
glance
throw
at
some
what
swing
the substrate or the seafloor might be in terms of its character. If it's highly reflective,
copy.
we get a high intensity return, or if it's absorbing or scattering the sound, then we get a low intensity of return.
It's okay for now, but if you see the tether getting close, let me know, and
Yeah.
you can go ahead and stow it at your discretion.
Okay.
And Derek said that in this area, it was sort of moderate intensity, meaning there was some sort of reflection, but also some sort of scattering and absorption, but it was very similar across the area, which completely makes sense when you see, from what we've been seeing, it's all very, very similar in terms of the substrate. It's this dead rubble coral that would tend to scatter some of the sound, but it's almost an armoring of the mound, so it would also be reflecting some of the sound. So that kind of explains maybe why it would be a moderate backscatter. And earlier we had Matt Pody and his group of mappers at NOAA in Silver Spring, and I don't know if he hasn't been in the chat room for a while, but maybe they can give us some information about this area if they have any.
And
See if I can't get out there.
we did
know if
pass
I'm
over
stretching out too
a
much.
Bathypathes black coral.
There's two
just
big
a little
bush.
bit ago. Ooh,
Yeah,
it looks
it's
like
another.
a
Yeah.
Liopathes here
Dark
on the
one's red.
right,
Yeah.
the
Dark color.
Very cool.
darker big bushy coral, and then maybe a bamboo coral here on the left.
Pretty large colonies.
Yeah.
Big, beautiful corals.
Must be some good solid substrate there.
Yeah, they must have something that they've attached to. Could be the dead coral matrix. Yeah, that does look like a big bamboo coral on the left there. That's beautiful.
We'll start with this one.
This is Liopathes, a black coral. And black corals are often colored, and so it seems counterintuitive to call them black corals, but if you were to scrape off the tissue, which is on the outside of their skeleton is
Okay, do you want to get
actually
lasers on for a few seconds?
Going on.
a black proteinaceous substance.
Sounds great. Thanks.
And it can be shined up, and
that
it's
good, or do you
actually
want- Yeah, go ahead and
harvested
turn 'em
for
off.
Alright,
jewelry
thanks.
in some places. Not necessarily this species, but
Should just have
black
them on, like, a
corals in general.
momentary.
All right, video at your discretion. You can go ahead and zoom in.
You can see the black inside the yellow tissue, the polyps.
You want to take some still cam shots?
Yes.
And the branches of this Liopathes are very light and feathery. It's a little bit difficult to sample, especially in a high current flow such as this, because as soon as you snip off a piece, it will float away in the current.
Yeah, you can see just how strong the
still
current
camera
is
might be
here,
shooting
which we've
over
been,
top of it.
you know, when we zoom in, really sort of
Maybe,
see the branches.
ROV nav bridge move.
And
yeah, that's true.
on that note,
Copy,
I
bridge.
should
Thank
introduce
you.
myself as Stephanie Bush from the
Ships
Smithsonian
just
Natural
stopped
History
moving.
Museum,
Copy that.
sitting in for Leslie as she goes off to lunch.
Do you want to go ahead and do another?
And it's Taco Tuesday here on the ship.
Another move? Sure.
Yeah.
Yeah. I think
20 meters.
I think so.
Oh, right away? Okay.
Thanks for the reminder that it's Tuesday, actually.
I know. I didn't know what day it was at all until they told me it was Taco Tuesday.
So we can go
Yesterday, I definitely thought it was Tuesday.
So do you want to change the heading to the north, co-pilot?
It's pretty great when every day you get to explore a new place. And
there's a nice urchin at the base of the Lyopathes. I don't know. Maybe you mentioned that and I missed it when we were switching.
Would you like me to zoom in on the urchin?
I didn't see that. Sure, if we could zoom the urchin.
What's up, Levi?
Sure, that would be great.
Your back left toe is in the sediment is all.
That's what's anchoring me down.
It looked like a, and you guys may have seen this earlier, but it did not look like one of the pancake urchins.
And we're also looking at
relatively longer spines.
Current shifting a little bit more to the
go ahead and do a quick zoom there.
Maybe thicker spines?
It would be nice if we
could
about
yeah,
5
like a pencil
degrees
urchin.
to the right at some point. Over.
We just saw a pencil urchin. Yeah. That looks like another pencil urchin.
So I missed
yeah,
that.
we'll do it right now.
I'm losing a video.
And then, did someone tell us what this smaller white coral is in front of the Lyopathes?
I imagine that's another bamboo coral.
To starboard, I'm going to come to 172, 5 degrees to starboard.
Okay, coming down to this one, Levi.
So Tina thinks that the large orangish
go
coral is
full wide
Lyopathes CF glaberrima.
Including the skids. Go ahead and go full wide.
Similar to that species at least, if not that species.
Turn slightly to port.
And so Scott France was saying that this might be the Clade D keratoisis that he was expecting to see here. What a big, beautiful coral this is.
Yes, center of rotation is A-frame.
There we go. I can see it. Tip of it in the still cam now.
And we're a little bit deeper again. We're at 700.
Fernando, can you
28 meters
zoom
or
on
so.
high pack to remove the screen so I can see the ship?
Go ahead.
Okay, thanks.
Sure. I'm going to set down in a second. We'll get a good laser shot.
Got lasers.
Thanks. How's that tether? It's okay, right?
Yeah, it's
and
it looks like there's a pancake urchin underneath this large bamboo colony.
Move over.
Right at the, almost right at the base.
Do you want to try the fog lights here?
And I hear that Chris Ma told us about pancake urchins when I was downstairs
Video, can I turn
eating
those
lunch.
forward up?
Great to hear he's back in the U.S.
Yep.
And hope he'll be joining us again often. And going through all of the images of sea stars that we couldn't identify while he was at a meeting in Japan.
Did you notice those lights help at all?
Oh, yeah. The still cam shot.
Pilot, this is watch too.
Go ahead.
Can we get a zoom in now that we've gotten done the lasering?
Yeah, we're getting a few shots with our still cam, and then I'm going to go ahead and zoom in for you.
Okay, cool. Thanks. Yeah, that was probably it.
Yeah, see that, Levi?
Yeah, I think there's a stylosterid
Yeah,
coral actually growing towards the base of this
try to get in real close
bamboo
here.
coral. That's the white hard coral you see in the bottom left.
Real nice high-res shots of those polyps.
And so Scott France is going to be looking at the branching pattern here, and he's predicting
full
that these should be internodal branching if it is the keratoisis that he thinks it is.
Okay, go ahead and zoom in video.
I'm kind of surprised, at least so far, I'm not seeing any associates on this one for such a large colony, but maybe we'll see them now that we're zooming in. Although we did just zoom past a branch that looks like it's lost some tissue, and I know in previous dives we were talking about
let's come out video.
Brittle stars or basket stars that maybe braiding the tissue off just by being present, not necessarily eating the tissue, but
light bar still
scuffing it off, if you will.
Yeah, my heading just ran
before we leave this area, could we also get the lasers on the Lyopathes orange coral to the right as well?
They did.
Sure.
Or did they do that?
I think they
to
did
the
that,
right?
yeah.
Okay, it was a request and I didn't
I think that was from a delay,
The big one, right?
Yes.
Yeah, we got lasers on that.
Great, thanks. And Tina Moldasova made a comment about the Leiopathes, and she said
I turned
that
to port to keep you in field of view.
as far as she knows,
That's fine.
There's
just let you know.
Not much genetic difference between the different morphologies that have been found so far. Well, that's very interesting.
So Scott's confirmed that this is this coral colony we're looking at right now is keratoisis, although he's saying that I don't know if that he's saying the internodes are quite short. I don't know if that's a comment about this particular one or that's how you tell that it's keratoisis. So sorry. Relative to the thin branching bamboo coral we were seeing earlier. Okay.
Okay, let's come out.
So looking at the slight differences in morphology, branching patterns and things like that, that allows us to differentiate between species.
There's that tether.
So it goes down on my screen, which is good.
What about,
I think it's
relative to you?
Pilot, let's watch two.
Way off to the side. Yeah. Go ahead, watch two.
Before we take off from this spot, they're talking about possibly collecting this coral, so I guess
the white one?
Don't go too far away until we can figure that out. Thanks.
Well, actually,
ROV nav bridge move complete.
All right, we got covered. This is not to be collected, so we can keep going.
Okay.
Thanks, though.
So we just finished that.
Look at all those cutthroat eels in the distance here.
Yeah.
Levi, you're happy with that?
Tether?
Pretty low.
So those last coral colonies we were just looking at, the keratoisis and the Leiopathes.
From my port up and then down.
Those were some of the larger corals we've seen today so far, correct?
Yeah, they really are.
Okay.
And I don't know if we're kind of on the side of one of these mounds. We've gotten a little bit deeper than when I left. We were at 700 meters.
Smack you here, right?
So yeah, we're definitely
we
kind
don't
of
want it to get over the top of Sirius.
The side that sort of is dropping off into deeper water, so that makes sense. That have a little bit more, even more current in this already fairly high current area, where we would see these larger coral colonies. And had we seen Leiopathes today in this dive?
Yes, we have seen at least one.
Okay, just not as large.
And that one was partially dead, and there were other organisms living on the skeleton that was exposed.
All right, so which direction do we want to go now? Do you think we've gotten over far enough?
Probably pretty close to north, I think.
Yep, I agree.
That's nice. Yeah.
Or is it going to start curving around to the northwest?
Yeah, maybe. Maybe if you take a peek out, see what the ground looks like.
I'd rather keep you kind of over the ridge more, because if you go out and then face toward me, like the current's going to be blowing directly into D2,
right? Now we're getting a good overview shot of the two colonies we were just taking closer looks at.
So your heading is 330.
The Leiopathes, black coral on the left.
So is 330 going to get us to
on the right?
Where we need to go?
Bearing is 335.
On the moves you've been doing?
Oh, no.
So you can see in this nice overview that they are kind of alone out here in this particular section of the mound, at least size-wise. There are, of course, some other smaller, looks like hard corals down here, but those ones are really growing up and out into the current flow.
We have been doing 015 so far.
Yeah.
So you're saying you want me on the ridge and then you off the ridge? Or do you want me?
Yeah, I think. Doesn't that make sense to you?
Yeah, that'd work.
Instead of, because otherwise you're going to have to turn the port. Yeah. Though I guess if you turn and get the tether on the other side of you, maybe that would be more ideal, right?
It kind of naturally wants to be on this side, though.
Yeah?
Yeah.
Well, I'm just saying if you get over far enough and then, like, even if you're facing, like, 90 degrees from the direction of motion, it would be blowing way off to your side, because you see how it is right now. It's kind of stretched out right in front of you.
Yeah. Okay, I see what you're saying.
You see what I'm saying?
Yeah.
What do you think?
So yeah, we can move Sirius west.
So right now the pilot and co-pilot and navigator are discussing which direction to go along this
So why don't you figure out a direction with nav?
Mound, and that's partly because of the strong currents that are occurring down here and in this region in general. So they have to do a lot of tether maintenance so that the